All right, so I think I'm going to get started. I'm Rachel Hostetler. I'll be moderating this discussion today, and today we'll be having talks featured from Janelle, Delissa, Michael, and Cindy. And today we'll be talking about spatial transcriptomics and how the Allen Institute uses team science to create large spatial transcriptomic data sets that are featured like the ones in the ABC Atlas. And as a reminder, this webinar is being recorded currently. And this is part of a webinar series. We have 7 other webinars after this one if you're interested in learning some more. The series will also be put up on our YouTube channel after the recording is finished. We will be taking questions during this webinar, so please post them in the Zoom. And if you're watching a recording of this after the fact, please post any questions that you have in our community forum, which we have posted in our YouTube description. And so we're at the Allen Institute, and the Allen Institute focuses on a few main principles. And those principles include team science, big science, and open science. And today's talk is specifically featured on team science. And now we have these large teams that work together to tackle these really large data sets and large experimental pipelines. This project is also an example of big science because it features data from many different sources and many data sets. And it also features open science because this data is shared openly for others to use in their own research, and it's shared freely online on brain-map.org. And over the last 20 years, the Allen Institute has done a number of large-scale projects, starting from the mouse reference brain atlas that was created almost 20 years ago, all the way to our Allen Brain Cell Atlas, or ABC Atlas, which was released in 2023 and features single cell and spatial transcriptomic data. And spatial transcriptomic data is what we'll be highlighting in this webinar today. And again, some of these data sets featuring our spatial transcriptomic data can be viewed online freely in tools such as our ABC Atlas or Allen Brain Cell Atlas. Here's an example of what that looks like. So after this webinar, if you're curious at using some of this data for your own research, here's how you can access it. And again, we really use team science to tackle these very large scale projects. So we have many different groups that are working together at different parts of this pipeline. And today in this talk, we'll specifically be covering groups from histology, imaging, spatial bioinformatics, and informatics and analysis. And now we'll be starting with our histology section featuring Janelle.
Hello, good morning, everybody. My name is Janelle Arisa. I am the manager of the histology laboratory at the Allen Institute for Brain Science. Next. So, as Rachel mentioned, we are part of many labs that are called the production labs. And that big effort requires many people. We are a part of those teams. Next. So we collaborate with several groups, and especially in several of the products that we release in our webpage. And we are part also of many collaborative projects like cell types and biogenetics tools. And today we are going to talk about this spatial transcriptomics. And we did this thanks to the effort also with and conjunction work with the animal services. Thanks. So we are a small team, but very powerful team. So you will see what is the high quality work that we generate every single day. So as I mentioned before, we work together with the animal services and the spatial transcriptomic teams in relationship with this specific mouse brain atlas. Next. So to get an idea of how we start the whole process of the mouse brain atlas production and analysis, we have to perfect every single part of the process. And everything is start with a high quality dissection process. So we partner closely and work closely with the animal services I mentioned before. And we had an agreement that they have to get us a perfect brain dissections. And when I say perfect, it's like almost perfect. As you may know, the brains that we use for this brain atlas are not perfused, are not clean with PBS or anything like that. So it requires that high precision dissection with a very mushy brain without fixation. So that requires that the brain is taken out of the skull very carefully to avoid any damage on the structures or the main structures of the brain. And we only process for these specific analyses brains that have no damage whatsoever. So they oriented in the OCT molds and gave us the specific orientation so we can get the specific coronal sections. We perfect the temperature sections and the crystal conditions to get the high quality sections. And we noticed that, for example, according to my past experiences, that if we section the slices, the brains from an orientation posterior to anterior, we can get high quality and very complete tissue sections. Then we dry the slice and we partnered with the engineering team to design specific racks to, for example, hold the round cover slips for certain platforms. And so we are able to process and keep consistent post fixation and dehydration process in PFA and ethanol. And then we design in-house also boxes that help us to keep these round cover slips in 70% ethanol storage. So then we can deliver it without breaking it or damage in any part of the cover slips and deliver it to the Spatial Transcriptomics team. Next. So, as I mentioned before, there are so many factors that need to be perfected. The perfect brain dissections, the custom brain freezing techniques that we have to keep to avoid freezing artifacts or freezing damage during the process, maintaining constant crystal temperatures that they have to be reliable, and they has to be consistent all the time, ensuring consistent high quality tissue sections. And that means that we always make sure that all our environments are RNase free. Cryostat solutions, brushes, everything that touches the tissue has to be super clean and RNase free. Also, we have perfected sections at 10 microns, 14 microns, and 20 microns for spatial transcriptomics analysis. And we have consistent sectioning and fixation protocols. To have an idea of how many hours it takes to section an entire mouse brain, for example, for the mouse brain atlas, it takes us like 200 sections. We collect 200 slides. And the average time that we spend sectioning is 9 hours. So we take two shifts to avoid the one person takes all the job, and by like being exhausted causes any problems during the sectioning. Also, for example, for the macaque, we take 100 slices and we take an average of 4.5 hours. For the human sections that we use for the HMBA project or the CAD atlas, it takes us like 3 hours to process one tissue block, and half an hour for the human Alzheimer's disease project. So the key of all this is hard work, consistency, and reliability. Next. So for the tissue, for the mouse brain atlas, we section two animals, two brain, the mouse brains coronally, and two were sectioned sagittally. The sections were made at specific intervals of 200 microns, resulting in 150 coronal sections and 25 sagittal sections. Next. For example, for the CAD Atlas and all the publications that we have done so far for the middle temporal gyrus, we processed 35 tissue samples in a very consistent way, as we did for the mouse brain atlas. And we are currently working to obtain the prefrontal cortex spatial transcriptomics data. So our current work also include the mouse postnatal spatial transcriptomics in P0, P4, P8, and P20. Also the develop of post-spatial transcriptomics, but is immunostaining to detect the pathological peptides in those spatial transcriptomics slides already analyzed. We are also creating the atlases for human, macaque, and marmoset basal ganglia using the spatial transcriptomics platform, and we are directly involved in these projects. So it has been like a year since we released the ABC Atlas, and we have been improving all the time and make it more specific, more reliable, and more consistent in every single step. That's why the scientists have created a specific sectioning guide that we respect at every time. Our engineering team have designed a specific camera that take a picture of the block face of the tissue block, and also take a picture of the microns, so we can reconstruct in the C-plane all the tissue sections that we obtain. We cut 300 microns off of the slab of the surface to make like an even surface and remove all the frosting damage. We collect in histology sections, and then we collect the merscope or CNU sections or stereosig sections. And we collect also the nasal, the sections that are going to be used for quality control purposes that we are going to stain with nasal and hematoxylin eosin. And if those sections or those tissue sections pass the quality control, we then deliver those sections to the spatial transcriptomics team. If they fail, we start the whole process all over again with another tissue block or brain. So when I mentioned that we collecting different histological slides is because we do also a different type of stainings on the tissue to assure the quality and make it easier for the complement the analysis in the spatial transcriptomic side. The histology team also collaborates with several other projects in the human, macaque, and mouse cell types project, where we do the processing of the tissue sections, and we stain them with the AP to make it easier for mapping posteriorly. Next. And also, we collaborate and make sure that all the tissue sections are also perfect for the Viral Genetic Tools Atlas that it has been released pretty recently. So now we'll leave you with Lisa.
Good morning, everyone. Good morning. My name is Alyssa McMillan. I'm the manager of Spatial Transcriptomics. A few years ago, I asked Michael to, who you'll hear from in a little while, for a talk title, and he says the Spatial Transcriptomics team puts the where to the what. And so that's basically our motto, and our mission is we are constantly putting the where to the what. We are part of this big pipeline. So we take in product in order to push out data. So I wanted to give you a little history about spatial transcriptomics at the Allen Institute. I want to take it wasn't started like a year or when the atlas came out. It was started several years ago with Jenny Close starting the spatial work at the Institute, and it has grown since then to the inclusion of my team as we've moved on through the years. One of the reasons I wanted to show you this brief history of the spatial work that we've done at the Institute is that we have relied on the past knowledge and the past challenges in order to build on what we have today. So with the development, one of the things that we have learned is that in order to create this spatial map that we're trying to create is that you need a well-specialized trained staff. Each RA is trained to work on all the platforms that we use, and they each bring their individual skills to different parts of the process. We are a cross-species, cross-platform, cross-institute team, which simply means that we work on a lot of different projects over a lot of different grants. I mean, we help many teams throughout the Institute, as well as our other scientific units at the Institute, whether it's to help them with knowledge base, to help with training or anything that they may need as we have moved on to optimize many parts of this process. So many of you know we profile using MERFISH. I think many people are acquainted with this method. So as well as with a highly trained force, you need a well-honed fleet of instrumentations. At the Allen Institute, currently we have 8 MERScopes, and we have 4 10x xeniums. So the MERScopes come from VizGen and the xeniums come from 10x. And this is our fleet that we do all of our spatial work on. Another thing that we like everyone to realize is that this process isn't a short or quick process. The planning for the MERScope and the xenium pipeline takes months of planning. It takes months of planning with the teams, preparing of samples with animal care. And then once it goes through histology has to do their work. And then it comes to my team. I like to call this, basically, this is a relay of trust from one person to one team to another as we move it through the pipeline until it reaches, Michael's team with the data process for him to do his part, they do their part. And it's a similar process with our xenium pipeline. One of the first challenges that we had to overcome was the enormous amount of data that we generate. So if you look at the numbers that I put here, so when we do a whole mouse brain project that we do, it was based on the 500 gene panel, and each one of those cover slips with one section generated between 3 to 5 terabytes. Since we have 8 MERScopes, we have a tendency of generating between 24 to 40 terabytes of data per day, which potentially is up to 160 terabytes per week, giving 640 terabytes per month of data. So that was the first thing we had to, how do we get all that data? The MERScope can only hold 14 terabytes of data at a time. And so with our engineering team, and you will hear this a lot, engineering team, they came up with their software engineers came up with software to basically pull the data from our platform, push it to AWS, and then later after all the analysis is done, it's sent to the Bill or to Nemo or repository for long-term storage. But it was crucial to figure out and work with not only our engineering team, but also with our platform companies in order to be able to do a lot of this work so we can pull this data out and keep the flow moving. My team, we imaged up to 28 samples per week on the MERScope and about 16 on the xenium. So we need this constant flow. And this is basically the flow with the xenium process. As I say, we do up to 16 runs a week, 2 runs per machine. So it's critical that we get the data moving quickly. So when we first started with the ABC Atlas, I needed a way for my team to take a look at to make sure that we were actually putting the where to the what and making sure we were getting things in the right places. So we picked out we asked the teams for laminar markers to take a look at where they should belong. As well as looking at the laminar markers, I then compared the laminar markers we were given to the ISH atlas of the same genes to make sure that we were getting them in the right, positioning. And then once we did that, we were able to say, okay, we know that those genes are in the right position. We could then move forward. As I said, we are a cross-species team. So we did this across all the species, making sure that we had those minor sanity checks at the beginning before we were able to put QC metrics to them. And we'll talk about the metrics that we came up with in a little bit. So the two of the places that we've optimized this protocol, and I think gives the greatest impact to the later product that we now produce, is in the embedding phase and also in the QC phase. Those are the places that we've really worked hard to optimize in order to get the best data output. So one of the problems that we had seen was this gel lifting. And so basically when we would see on the actual gel before we image, we would see these small bubbles. And these bubbles would then lead to missing areas of transcripts on the imaging. One of the ways you can detect this, when you look at the DAPI staining, you'll see the blurry regions. So a metric came up in order to detect basically what percentage of gel lifting. And this percentage, and as the higher percentage goes up, it informs later on down the pipeline if this section is usable for further analysis. Another problem that we saw going for is the Z-plane dropout. So as you know, it's the MERSCO sections, images in various planes, and we have 7 planes in which it images on. We saw this dropout happening where at the surface level of imaging, it would have these high transcript counts, and then as each plane goes up, we would get this drop off of transcripts. So what we did was over time, and as you see, nothing is done with a quick rash change. Over time, we went in, we looked at different sectioning sizes. So we looked at 10 microns, we looked at 14 microns, we looked at 20 microns. We wanted to see where we could what we could change in order to do a better job of reducing this Z-plane dropout. And as you see, with thicker sections, we were able to reduce that severity of that dropout. So we have transitioned, and we did try it in many species, and all the species that we worked in order to see how well it worked. So we tried, and this is the 14 microns. So what we have done was that we have switched to the 14 microns for most projects, and there's a few projects where we are sectioning at 20 microns. And as you see here, the numbers that goes with that. So it's not just us looking at it visually, but we have numbers in order to prove that the change was necessary. And here's the Z-plane comparison in here and the numbers. So transcript density is very important. And so we monitor that all throughout the process. Another thing that we found was this checkerboarding pattern that was another problem for us. And so we had to basically detect and also put that into a part of our metrics and see how that affects our data. So in comes two of my arrays along with our director, Jack Waters. So Naomi and Paul and Jack worked together to come up with this basic QC package, which we call Merquaco. So Merquaco takes all of these seen problems that we have or seen challenges and basically quantifies them. And so by running every section that comes off of all of our machines through Merquaco, we're able to see and measure and compare based on historical data where each section falls. So we'll know if we're having a series of something with higher periodicity, or we're having something with higher transcript dropout or FOV dropout. We're able to do a comparison using Merquaco that was developed. So by the numbers, as of beginning of October, on the MERScope, we have imaged 1,583 sections, which equates to 1,320 cover slips. On the Xenium, we have imaged 365 sections. In summary, that during 2022, the Allen Institute established the pipeline for spatial transcriptomics using MERScope, which we are now increased up to 32 sections per week. We continue increasing our capacity, and we're hoping to increase our capacity even further with tweaking of the bench work. The current team, made up of Naomi, Paul, and Jack, developed the QC package on Merquaco. Our scientific teams led by Michael, Brian, Jenny, along with their data analyst, Emily, have developed pipeline for filter mapping and alignment, and they improve overall our understanding of how transcriptomics identity relates to spatial location. And we have successfully generated several papers using the work that we have done in our team. So thank you. Oh,
all right.
you got it?
Okay, I'm going to start sharing into presentation mode. All right. So hello, everybody. So as Delissa already mentioned me, I am Michael Kunst. I'm a senior scientist here in the imaging department, and I have the pleasure of taking the data up after all the hard work is done and then just start applying some computational tools just to make them look even prettier than they already are. So I am part of a broader group that we are loosely calling ourselves the Spatial Bioinformatics groups. So as with everything at the Allen Institute, none of this happens in a vacuum, and there are lots and lots of people involved in every individual processing step. So yeah, just a few members of that team. But so just to briefly go through the workflow. So it starts off by the process of segmentation, where we assign individual mRNA molecules to cells so they become amenable to single cell analysis tools. Then we do a first broad filtering step to get rid of the low Q quality cells, or at least the obvious low quality cells. And the next step then is to assign each spatial cell we have its own identity based on its gene expression profile. And we do that by mapping it to a reference taxonomy that has been developed using RNA-seq. And then the last step in the processing part is we then take these brains and try to register it to our common coordinate framework, which then gives us the ability to assign individual cells to specific brain regions, but also to integrate it with other data, such as single projection neuron data that has been all registered into this same standard brain reference space. Once all that is done, the fun actually starts. We can start doing more high-level analysis of the data and trying to understand the composition of cell types in the brain. All right, so like I said, the first step is segmentation, and I cannot stress how important that is, because in this case, what we end up getting primarily first is a section in which each mRNA transcript has its own location. So I just made a zoom out in here. So I labeled these are like all the individual mRNA transcripts we can measure at an extremely high spatial resolution. But in order to apply single cell analysis tools, we need to assign these properly to the individual cells. And once you start making mistakes there, you end up with contamination that can have huge ramifications on any of your downstream analysis. So yeah, so it's really important to get this step here to this step here. So what I have shown here is the same cells, but I assign each transcript to its own cellular identity. So the question is, how do I get to that point? So we're using cell segmentation tools, and these rely on references that can that help them to identify the outline of a cell. Standard references for those are image stains for those are DAPI. So DAPI stains the nucleus. And then what we also get offered in the MERScopes is a stain for poly-T. So poly-T just binds to the poly A tails of mRNAs, and in theory should label the entire complement of mRNAs you have in your cell, which then should nicely fill out the cytosolic area of the cells, providing two nice background stains that you can use to define the outline of the cell. However, we noticed early on that actually poly-T is not of the original DAPI stain. And if you compare it to the mRNA, you can see that the cells actually are much, much bigger. So one of the first steps we developed a tool where we actually take the mRNA locations and turn it into a low resolution image at the same level as the DAPI and the poly-T. And we use that as our cytosolic stain. Another problem is that, so microscope experiments are done in 3D. So we have 7 optical planes to cover the entirety of the 14 micron sections. What the current and what the old segmentation we have been using so far for the ABC Atlas does is it actually only does the segmentation on one plane to save computational overload, and then applies it to the planes below and above. So it just picked out 3 out of the 7 total optical planes you can see. And that comes with a lot of problems. First of all, it assumes that cells are cylindrical. So you just end up with like an overestimation of cell sizes in the planes above and below. And we also noticed that even in the in-plane sections where we would expect a proper segmentation of the cell, that the segmentation overestimated the cell size. So what this leads to is you end up with low correlation scores. So how did we go about it? So we're using a machine learning tool called CellPose. It's a graph convolutional network. So we did 3 main improvements in our system. So the first one I already mentioned is we starting to use the mRNA signal and turn that into an image and use this as the cytosol stain. The next step is that so far we have been using one of the general models that has been provided by the software. So we went in and actually retrained the model with our own Merscope data. So the way it works is we use CellPose 2.0, which has this human in the loop system. So you start with the original CellPose model, you give it a new image, you let it do its is exactly what you need, and you don't need to do any more fixing. On top of that, we are now rewrote the code to have now a tool that lets us do the CellPose segmentation in 3D instead of 2D. So we don't have the same kind of cylindrical cell shape anymore than we had before. And just to look at this, so here's an example, and I think you can already immediately appreciate the difference in the cell segmentation quality here. It's really good at detecting cells in very dense areas. It also much more stays around the actual cell outline. And you can actually see how cells change in size if you go through the different Z planes. So this one here changes. So here I plotted the distribution of a couple of our main QC metrics. In greenish, you have the distribution of the new segmentation, and in oranges, you have the distribution for the old segmentation. The first one is cell volume. It's pretty obvious that the cell volume has been much more decreased in our segmentation, which is what we would expect, since one of our main concerns of the original one that is way overestimating the cell volume. So that's good. We also looked at the number of detected genes. So we are limited in the number of genes. So we only have 500 genes. So our panel is designed to be very specific. So that means we don't have a lot of genes per cell. And in this case, if you increase the number of detected genes, that normally is an indicator that you pick up something that is from a different cell, since they generally have very complementary gene expression cells. So again, what we wanted to see is a decrease in the overall number of detected genes. Same is for the detected mRNA. Just by the sheer fact of the bigger you make your volume, the more likely you should pick up stray mRNA. So seeing a decrease is a sign of increased quality of the cell segmentation. And then reverse is we actually see the density of the mRNA go up. So if you looked at, if you remember the earlier like cylindrical cell shapes, you saw that the area where the actual mRNA is decreases the further away you go from the cell body, and that extracellular mRNA has a much lower density. By not including all these low density areas, we increase the overall density of mRNA within our cells. And lastly is we made a list of what we call incongruent genes. So these are genes that, based on our RNA-seq taxonomy, we know should not be expressed in the same cells. So what I plotted here is then the distribution of percentage of incongruent genes per cells. And you can see that the distribution goes significantly down, meaning that we have less genes found in cells where they're not supposed to be. So overall, we just tremendously increased our segmentation quality. So that's the first step, and this is very crucial because this sets the tone for the rest of the analysis. Because if you don't get this right, there's no way you can fix that later on. So, okay, once we have done that and we put a lot of effort into increasing our segmentation, we still end up with a few low quality cells. These could be for various reasons, tissue quality, or just you can end up in a way that you section your cell just like pretty much in the middle of the cell, so you only get a partial footprint of the cell. So we then have to come out with ways to measure and verify low quality cells. So, and in this case, it actually helped, two things helped. One is we can look at the distribution of cells in the tissue, since we're using spatial transcriptomics. And we also have certain areas where we know that some tissue problems arise from our earlier data. So we can start around plotting the distributions of all these different values. So we again looked at the number of genes detected per cells, number of mRNA spots detected per cells, and then also blanks, which I come to in a minute. So we found out that if we filter out cells that have less than 6 genes detected, these strongly correlate with the regions where we know there's, for example, gel lifting, or some other problems where we saw that the detection of genes is a little bit reduced. And the same is done for spots. So in this case, we can see it's strong in certain areas where there's clearly not enough mRNA detection, but it also very clearly labels the areas around the gel lifting. And then the last one is that I look at what's called blanks. So blanks are basically a measure for false positives. So if you're familiar with MERFISH, you know that genes have these barcodes, and they have a certain Hamming weight and Hamming distance, so you have them on in 4 rounds. So blanks are basically follow the same logic a gene follows in a code book, but it does not encode for an actual gene. So every time you see a blank, that is just basically a sign of a false positive detection of the whole system. So we found in regions there the blanks are higher than 2% are also strongly correlate with low quality tissue regions. So we use that as our next filtering criteria to find bad cells. Okay, so much then for the filtering. The next step is then, almost done with the filtering, is the doublet detection. So just like in single cell sequencing, where you can have 2 cells in 1 droplet, you can have doublets in spatial transcriptomics. This happens mostly when you have cells that are very close to each other, and the cell segmentation algorithm is not capable of differentiating between those 2. So how do we find them? So we used a tool called Solo. It's a deep learning approach where you take your data and you just generate artificial doublets by randomly taking 2 different cells and merging them into a doublet. You then encode everything into a deep neural network using a variational autoencoder. And that learns a classifier that then can be used to run over your data and identify which cells can be classified as singlet versus doublet. And again, we have then we have to play around with finding the threshold and which to set of a singlet or a doublet. And again, we have the advantage of we can actually look at the data and visually verify, do we have a singlet or do we have a doublet? So in red, you can see the cells that have been identified as doublet. In most of the cases, you can clearly see that there are two DEPI nuclei that are merged into one cell. Occasionally, there's kind of like a border case, not quite sure if this is really two, but you can clearly see these are two, whereas the single ones are obviously identified as singlets. We can again quantify using certain measures. So again, we can look at the number of genes detected, much higher in the doublets. Again, the way it works is that genes of the panel are highly expressed in specific cells. So if you have two different cells, you end up with a lot of genes that would be considered incongruent. So you just end up with the overall higher number of genes detected. Obviously, the cell volume is bigger in a doublet because you merge two cells together, as well as the number of spots detected. And again, the number of incongruent genes, it's a bit hard to see, is severely reduced in the singlets compared to what has been described as doublets. Okay, so at this stage, we got rid of the most obvious low quality or doublet cells. So the next step is then to assign the cell type A identity. So for this, I have to step back for one moment. So something we haven't talked about here yet is this whole project is centered around a RNA-seq taxonomy. So there has been a lot of effort put into generating this taxonomy where RNA-seq data has been collected from multiple mice, from multiple brain regions, and has been clustered using advanced bioinformatic tools. And we ended up with a taxonomy that consisted of about 34 classes, over 300 subclasses, 1,200 supertypes, and over 5,000 clusters. So it's hierarchically organized. And we could already see that there are region-specific cell types. But the idea with spatial transcriptomics is that to have a bit more of a fine-grained anatomical resolution in figuring out where these cell types are. So how do we map the spatial cells to our RNA-seq taxonomy? We're using a tool that's called Map My Cells. It's also developed here at the Allen, is available now as an online tool. I keep forgetting to put the link in here. I'm sure somebody can put it in the chat. And the main idea by that is that first, you take your individual Meriscope cell, you have its gene expression profile, and then you compare it to the median expression profile of a cluster. And we do this in a hierarchical, so we do this, you take the cluster median of you take the gene expression median of a cluster. So what you can do is you take each cell and you do it compared to every individual cluster you have in your data set. And then you do this 100 times in a bootstrapped way where you only use 90% of the original marker genes. So you end up with an average correlation score and you can assign a cell to a cluster. So you can either do this in the flat way, so you take each cell and compare it to each cluster, or you can do it at a hierarchical level. So as I mentioned, there are multiple levels of hierarchy. So you start with the first one, the class. And then in this case, you don't cluster by, you don't take the median by cluster, but you take it by the broad class. But you also don't take all the genes that are in common between the Meriscope panel and the RNA-seq panel, but you only take markers that are defined as markers between the individual members of the class. So in this case, you would assign this cell to the IT-glued class. So then you go to the next step, you go down to the subclass, you do the same approach. But in this case, you only compare the cell to all the members within the class and not any other subclasses that are in other classes. You again subset it by the marker genes, you run the 90% bootstrapping 100 times, and then you assign it to the next level in the hierarchy. And then you can go down to supertype and clusters and end up with a cell type assignment. So ultimately what happens with that is you start from these unlabeled cells of this coronal section here and can assign them all to the individual. In this case, I labeled them by subclasses. And you can already appreciate the high spatial regional specificity of individual cell types. You have differentiation between the medial and the lateral habenula. You have the different CA regions of the hippocampus. You find individual layers of the isocortex. And you can even find, if you squint hard enough, individual nuclei within the hypothalamus. Okay, so now we assigned this. We're still not fully done with cleaning up the data set. So as I said, the outcome of this is an average correlation score for each cell within a cluster. And now we have to still deal with the fact that there are probably some cells that somehow survived our initial filtering step. And these normally show up at a low correlation score. So one way to go about it, and what you initially did, is you could just set a fixed correlation score, and everything what we said was below 0.5 is going to be removed. However, there's some issue with that, is that the median of the average correlation score for cell types can differ wildly between 0.3 to 0.6. So setting a fixed threshold is a bit overly reductionist. So what instead we did is we calculated it dynamically based on the distribution. So what we calculate is the median absolute deviation in both directions, like for the upper and the lower one. And then we take 2 times the lower median absolute deviation and set that as a threshold. So in this case, you have this example of this hypothalamic cell type. So we now set the threshold here. We filter out 19 cells. And once again, we can look at where the cell types are located. You can see everything in black is something that survives filtering. Everything is red is something that gets filtered out. And we can clearly see that these are cell types that are nowhere near the hypothalamus. It gets a little bit more complicated when you have a large number of badly mapped cells within your distribution. So you end up with something like this that looks like a bimodal distribution. If you would apply the double mat, you would be way too lenient on where you filter out the cells. You would only filter out one cell. So the way we go about that is we first identify cells that are bimodal based on comparing the potential peaks, then just basically cut them through the trough of the distribution, and then we apply the median absolute deviation threshold again. So this is what you see here in the lower set of cells. So, and again, you can see that the cells we find are not only near the not near the hypothalamus, they're also associated with low quality regions. So now we are at a stage where we are reached probably a stage where we are done with our filtering and we have a high quality data set. The next step is to then assign it to brain region by registering it to a common coordinate framework. So the way we currently are working on this is we're using a tool called Starliner that generates spatial domains based using a graph attention autoencoder. In order to feed our data into Starliner, we first actually have to take the original spot table and grid it into 50 by 50 microns and then sum up gene expression within each of these grids. It's kind of like a pseudo-Visium experiment with a much lower gene count. So we then use Starliner to generate spatial domains. One of the advantages of Starliner is that it's capable of integrating domains from different sections. So we can feed in the entire data set, and the spatial domains it finds in one section will also be labeled as the same cluster in the following sections. So once we've done all this, get it in our latent embedding, we can then run clustering algorithms such as Leiden and identify spatial domains based on that. And you can already see that a lot of these spatial domains reflect known anatomical boundaries. You can see the inferior colliculus here. You can see like parts of the hippocampus. You can see parts of the thalamus here. You see the striatum. So in order to register to our common coordinate framework, we decided to do a registration based on the broad region labels we have. So we then have to manually annotate these regions and send them to broader regions. So you have the cerebellum here, you have your medulla here, you have your midbrain here, you have the hypothalamus showing up here. And then the next step is to find a matching CCF section. So as you can see here are then the matching CCF sections. And it does a decent job, but it's not perfect. You can see clearly there are some regions that are not being properly captured. Like we clearly didn't have the inferior colliculus found in here. The pontine gray is underestimated. One of those reasons is that when you do sectioning, it is incredibly hard, if not impossible, to find to have an orientation of the section that perfectly matches like a perfectly horizontal sections you can find in our atlases. So the next step is then just to go in and manually adjust the tilts so that we have a much better overlap of the regions in the CCF. So once you've done that, the last step then is to adjust for any kind of individual variability and differences here by using more finer landmarks and using a non-rigid registration. So we go through that iteratively. So we go from interior to posterior if you find landmarks for all these various regions, and then do a more fine-grained registration to make sure that the cell types and landmarks get moved and properly overlap with what we see in the CCF, which then gives us now the ability to quantify these per region, but it also gives us the ability to visualize them in 3D. Here's just one example of how this looks like. I have picked two broad classes, which are the TH glut and the hypothalamic GABAergic neurons, and you can split them up in subclass and supertype. And in addition to seeing them nicely in 3D, you can also see that if you go down the hierarchical tree, you can see that the expression pattern or the localization of the individual classes on subclasses and supertypes becomes more and more refined, and becomes more and more regionally localized. Okay, so this is then the data that has been fully processed, and this is the moment where I'm going to hand it over to Cindy, where she can talk about what you can then do, what kind of high-level analysis you can do with these sets of data.
Okay. Yeah. Good morning, everyone. My name is Cindy van Veldhoven, and I'm an associate investigator at the Institute. And like the people before me too, I'm part of a very large team of people that analyze data that's coming out of various different pipelines, including the spatial transcriptomics pipeline. As Michael mentioned before, we have created a very high resolution cell type atlas using single cell RNA-seq data together with the MERFISH data to try and identify all the different cell types that exist in the brain. On the left-hand side, you see a representation, a human representation of the single cell RNA-seq data together with a constellation plot identifying all the different subclasses. And on the right-hand side, you see the MERFISH colored by these subclasses. And this is actually a very good start as to how we are using this data. So an example here is showing different neighborhoods that we've identified in the single cell RNA-seq data. This is the glutamatergic, mostly cortical cell types, the GABAergic subthalamus cell types, and the hypothalamus cell types. And in the single cell RNA-seq space, we have some information about the dissections that we've done. So we can place like very broad regions or attach very broad regions to the different cell types. But with the spatial data, it's possible to actually put a very specific location to it, thanks to the work that the teams before me did. So some examples of like these neighborhoods plotted onto the spatial transcriptomic data, you can clearly see that each of these neighborhoods has like a very specific domain that they occupy. Another way that we use it, and this is also a really nice sanity check, is to look at the correspondence of the transcriptomic identity and the spatial specificity. So on the x-axis, you have all the different subclasses that we have defined, and on the y-axis are the different brain regions. And there's a lot of different details in this plot. What I hope you can get from this is that Each of these larger groups of subclasses has a very specific localization. For example, these are all the cortical types, and they are mostly located in the cortex, olfactory bulb, hippocampus, etc. The same for the hypothalamus types. They also have a very specific location. With the exception of all the non-neuronal types, where most of these are present throughout the entire brain. So one way that we use the Merfish data extensively is to annotate our cell types. So using the single-cell RNA-seq data, we defined a hierarchy, purely computational. As Michael mentioned, we clustered the single-cell RNA-seq data to define a sleep level using high-end bioinformatic tools. And then we kind of cluster the different clusters back up. So subclasses and classes are defined by clustering clusters, and the same with the supertypes that we've identified, all the different levels. And each of these levels that we identify, we fine-tune kind of what the borders are using different versions of UMaps and very specifically the Merfish information. So for example, this is a cluster UMap for one specific group of cells. And we can turn this into a constellation, which is essentially a summary of the UMap. Each node represents a cluster. The size of the node represents the number of cells that were in that cluster. And the links between each of the nodes shows how connected or how similar the different clusters are within the data set. So we start with kind of this blank looking constellation. And then when we define the hierarchy, we can draw these different bubbles on top. But to identify, like sometimes when we do this kind of clustering of clusters, there are areas that are a little bit inconclusive. So for example, this type specifically in the mint green color was annotated, was split across three different larger classes or subclasses, which are the bubbles behind it. But then when you look in the Merfish data, you can actually see that this mint green type resembles both of the other types by occupying the same location, but based on gene expression patterns, they are different enough to classify it as a p-Phelp supertype. So this is an example of how we use the Merfish data in annotation of cell types. Other ways that we use the data are shown here. Like for example, this is a similar constellation plot as in the previous slide, but now it's specifically looking at pairs of types. So this is showing the MSN types in striatum, D1 and D2, and it's looking specifically at pairs of D1 and D2 types. And then we've identified a few types, but then when we look in the Merfish data, we actually can see, like for example, this purple one, this pair resembles this pair, and they are identified, or they are assigned to different subclasses, but they occupy the exact same kind of domain within striatum, and they share a large part of their transcriptomic signature. The other thing that I hope you can appreciate from this plot is that there is some kind of mediolateral to dorsal ventral organization of cell types. And we were wondering if this was driven by specific gene expression gradients. So in order to kind of uncover what these gradients are across space, we performed an independent component analysis on the striatum D1 and D2 classes separately in the single-cell RNA-seq data. And then each independent component is projected onto the Merfish data. And for the components with a meaningful distribution, we can start to look at the underlying gene module. So here are some examples of seemingly meaningful gradients or independent components. Now, when we look at the genes that are underlying these components, you can definitely see that is some kind of gradient across the different cell types that we've identified. So the dot plots are purely based on the single cell RNA-seq data, the same as these UMAPs. On top is that same gene module colored onto the UMAP in the single cell RNA-seq space, and the same gene module painted onto the Murphy's data. So this helps us understand how different spatial gradients exist within our data set. We can do a similar thing for the cortex. We've identified, or we saw that there are many supertypes that are organized in a very specific manner along the cortical depth. And we were wondering what are the genes that are driving this distribution. So we took a very similar approach as for the D1 and D2 types. And we can see that, or we can identify a series of genes that drive this distribution from deep to superficial layers. And the interesting thing is that these genes are not necessarily the marker genes that we've identified that represent either the clusters or the supertypes or subclasses. Another example is the organization of types within striatal amygdala and the bad nucleus of the stria terminalis. This is a view of a very complex class, what we call the CNU hypothalamus anterior class, CNU cerebral nuclei. There are a lot of different types that are not specific to any one nucleus, but tend to be distributed among different brain regions. And when we look specifically at the connection between the CEA within the striatal amygdala and BST, so the CEA the BST is a major output region of the CEA. So it is very interesting to see that many of the same cell types are represented in both of these regions. And one thing that is kind of noticeable is that there are specific domains that each of these types are present in both CEA and BST. So to dig a little bit deeper into what the organization of the CEA outputs in BST is, we performed spatial domain clustering, as Michael has mentioned before. And then by performing that, we can identify different subdomains within BST that are definitely linked to different CEA outputs. So these are the transcriptomic subclasses, and these are the spatial domains. Some are very strong, some are a little bit more mixed. But it seems that within BST, there are specific subdomains in which CEA outputs are organized. And then one final example is how we can link projection patterns to transcriptomic cell types. In this study, we injected a retro-AAV virus into the spinal cord of mice. So the virus is taken up by the projections and cells within the brain hopefully light up. And so we isolated the fluorescent cells from the different brain regions and sent them for sequencing. And we made a specific cortical spinal neuron taxonomy, and we integrated this new taxonomy with our Murphy's data. And this helps us to identify or to map the spatial transcriptome or the corticospinal cell types onto the spatial transcriptomic data. At the same time, the team used the same viruses in specific Cree mice and sent them for imaging, so we can validate the specific cell types in both the imaging data as well as in the spatial transcriptomic data. And with this, I'll hand it over to Rachel.
I just want to hop in while Rachel is coming. I forgot to mention the one way we optimized for gel lifting was by changing our embedding method, which was we went from the standard VisGen approved method to an inverted method of embedding by flipping over the cover slip onto the gel on a larger cover slip. Just wanted to make sure I made that clear.
All right, I'm just going to release a quick survey. If those in the audience could just quickly fill this out, this is helpful for us to understand how these webinars are being received and help us plan for future ones. So if anyone on there could fill that out, that'd be greatly appreciated. And then while we have this poll up, we are a little bit over schedule, but we do have a minute or two if there's any questions that people have. If not, we can jump ahead. Okay, I'm going to wait 1 more minute on the feedback survey before I close it. Okay, thank you for those who have participated in the survey. I just want to highlight a few other things real quick before we end the webinar. We do have other resources online on our website. So if you have more questions or would like to read more about some of the techniques that were talked about in today's webinar, we have a supportive resource catalog, which is posted here on the left. If you are interested in accessing some of that spatial transcriptomic data programmatically, we have some Jupyter notebooks to assist in that. And we also have a community forum where many of our scientists answer any questions that are posted on there by the community. And again, this webinar is also part of a webinar series. So this was the first one of this series, and we have some other ones planned throughout the year. We also have a workshop that will be at the Allen Institute, which is focused on cell types specifically and looking at transcriptomic data. And that will be in March of 2025. And that's primarily geared towards early career scientists, so mainly graduate students and postdocs. And we do provide a travel stipend for that. And more information on all of these events and other events we have is posted online at alleninstitute.org/events. And if we don't have any other questions, then we can say thank you. Thank you to all of our panelists. And I hope you all enjoyed this. And thank you. We will end this.